So, hi, I'm Kimberly Gray. I'm a professor in civil and environmental engineering, and I am joined by 4 PhD students in our program who are going to share with you highlights of their work. So we have Jun Kim, Natalia Obzut, Jen Chen, and Sam Wallace. And we want to share with you the really exciting work that our PhD students do as a testament to what we believe in among the faculty, that we're only as good as our students. And I think after seeing these presentations, you will agree that we're very good. So on that note, what we're going to do is Jun Kim is going to go first. We're going to go in alphabetical order. The students are going to give you, these PhD students have been here, I think everyone's been here, they're in their 4th year, so they're coming up on finishing probably within the year. Natalia's in her 3rd year, so she has a few more years to go. So Jun, you go first. You want to share your screen, and we all want to make sure we can see it.
Okay.
And everyone will introduce the group that they're working in and their professor, as well as, you know, the highlights of their research.
I'm sorry.
There you go. Perfect.
Okay.
Thank you.
Hello, everyone. My name is Jin Kim. I'm a 4th year PhD student, and I'm in Dr. Neil Blair's lab group. And in our lab, which is carbon biogeochemistry lab, as you can see from the name of our lab, the big theme that we're interested in is the carbon cycle and the human impact on that. So increased soil erosion, increased emission of carbon dioxide, they are well-known outcome of the human activities on the carbon cycle. And the vertical movements of carbon are relatively direct and evident, so widely studied and well estimated relatively. But compared to that, the lateral movement of carbon through the landscape is more complex and has not been fully understood. So the questions we have about that is like, has the land use change affected the carbon fluxes, and how they affected the routes of the carbon movement, and then what's the implication of such changes in terms of like global carbon cycle and further climate change and global warming and other type of environmental issues. So to address that question, our lab uses a range of analytical tools like elemental analysis, isotope measurement, and FTIR for functional group analysis, pyrolysis GC-mass, or GC-mass for molecular analysis. And using that kind of analytical tools, that I am currently working on understanding the dynamics of particulates in rivers during storm events. So rivers play an important role in connecting different places in environment. So they carry a significant amount of organic matter from uplands to the oceans. And that makes the rivers important pipes where their roles as a transporter is emphasized. But besides the role as a transporter, they are also important integrators and reactors. So they integrate the organic matter from various different sources, and also they provide a space where like primary production and chemical reactions can occur. So storm events specifically are the specific time where we can see the transition of the roles of the rivers. So with precipitation and the increase of stream discharge, organic matter from various sources is mobilized and introduced into the stream and further transported downstream. So my research involves assessing the biogeochemical changes of the sediment compositions during storm events so that we can better understand the roles of rivers as well as hydrological events on the dynamics of carbon. So the specific approach that I use for my research is biomarker analysis. So biomarkers are the compounds that have specific biological origins. So the presence of this certain compound can tell us about where the organic matter might come from, and also that can further imply that what environmental processes are associated with the different source mobilization. So far we have identified, so using this biomarker analysis and also other types of analysis, we have identified various sources in our study site and also their different timing of mobilization during storm events. And we're still looking into other storms to figure out what other environmental parameters might affect the different source mobilization during storm events. So here at Northwestern, I really appreciate the chance that I can get my hands on the real environmental samples and also the opportunities that allow me to think about what's really happening in the real field. And I think that's a really important training for environmental scientists. And I hope you can get that opportunities too. Thank you.
I always have to struggle to get down to my reaction button here. So thank you so much, Jim. That was really good. Thank you. So Natalia, are you ready to go?
And hope everyone can see it. So my name is Natalia Obschut and I work with Dr. Gray. And before I get started, I just wanted to thank you all for joining us virtually at Northwestern and share some pictures of our beautiful campus during the fall and our beautiful Lake Michigan and how it freezes over during our not so mild winters. But Dr. Gray and I are working on the concept of a lignin biorefinery and recovering resources from biomass waste. So this is the general schematic of our integrated biorefinery. And I know it's a lot to take in, but I'll go over this piece by piece. But kind of the big takeaway of what we're doing is that we're taking multiple streams of waste, including biomass waste, and we're recovering the resources from it. And it's usually in a green and mild way. And then in turn, these resources can be valorized into various products. So the first part of our biorefinery is the lignin. So what lignin is it is one of three components of a plant cell wall. So we have lignin, cellulose, and hemicellulose, and lignin is the second most abundant biopolymer on Earth after cellulose. And if we take a closer look on the structure of lignin, it's a bunch of aromatic rings that are linked together. And lignin is actually the only renewable source of aromatics on the planet. So then we ask, is there value in lignin? We know that we have a high production volume of lignin. We see some waste agriculturally from corn stover, industrially in the paper industry, and municipally in lawn clippings and newspaper. So we have the lignin there, but is there value? And there are different, there's a different degree of value that we can get from lignin. And we're kind of focusing on this very high product value. So this is the carbon materials, fine chemicals like vanillin, and ultimately pharmaceuticals. But we're not the only ones that have tried this. People have been trying to get products from lignin by depolymerizing it or breaking it into smaller pieces for over 50 years. But usually the conditions are harsh. They require a high temperature, high pressure, and they get these smaller products such as methane and carbon dioxide. Where we want to be is to get a high degree of functionalization while we're at mild conditions. So low temperature, atmospheric pressure, and without using harsh chemicals or expensive catalysts. So the next part of our integrated biorefinery is adding the waste effluent from a microbial electrolysis cell. So you can see a little microbial electrolysis cell here. And what that does is it takes wastewater with a high fatty acid content and treats it in the anode with the use of microbes to produce clean water. And then on the cathode side, we see that it also produces this caustic effluent that's often regarded as a waste, and it's high pH, high salt content. And we found that we can treat it with our lignin at room temperature and atmospheric pressure, and we solubilize it into a couple product streams. So what this looks like for us in lab is just a beaker filled with lignin, adding the effluent, and then we have this solution of a soluble fraction, and then this suspended fraction, which is also what we call lignin nanoparticles. So it's a very simple lab approach. And speaking of products, the third part of our integrated biorefinery is producing these precursors products. And we have our two streams, one being lignin nanoparticles and the other discrete aromatics. So what I spend most of my time working on is kind of identifying and quantifying what these products are. Luckily, characterization is made easy at the many core facilities at Northwestern. So a core facility is just a shared facility that houses the state-of-the-art instruments. So for example, for the suspended fraction of our solution, I have used the Shine core facility, which houses the nanoparticle tracking analysis, which helps me get a size distribution for our nanoparticles as well as the concentration. I've also used Nuance, which houses a scanning electron microscope. So from there, we can visualize our lignin and also see the shape of it, as well as a zeta sizer, which measures the zeta potential, which kind of gives us a stand-in for the stability of the particles. On the other side, the soluble fraction, I've been working mostly with IMSERC, where they have mass spectrometer and UV vis spectroscopy, and using the mass and the UV fingerprint, I can identify what kind of aromatics we have. And besides just the access to these facilities, the technicians that work with these facilities are amazing. Dr. Ben Owen over at IMSERC spent many hours with me, helping me analyze and sort through the data. And Dr. Nathaniel Cabot over at Nuance helped me prep my sample and kind of come up with the parameters so that when I go to use the instrument, it's made a lot more simple. That we look at is the product potential of what we are making. So with our discrete aromatics, we see that there's a various amount of properties and uses, such as flavoring, different precursors, and then all these properties that seem like it would make it useful for the pharmaceutical industry. So some examples of what we see is picoumeric acid, ferulic acid, and vanillin. And I'm sure you've all heard of vanillin, which is often used as a flavoring. Some of these larger aromatics are in the class known as flavonoids, and here we have trypsin and antirone F, and these have all of these properties, which makes it a good candidate in the pharmaceutical industry. On the other hand, our nanoparticles also have very interesting properties such as antioxidant, antibacterial, UV shielding, mechanical strength, and water resistance. And these have a wide array of different applications, notably in drug delivery, in the cosmetic industry as sunscreen, and also agriculturally as a way to transport pesticides or fertilizer in a way to minimize contamination and leaching. So it's important that we are recovering these products from a renewable biomass that's often regarded as waste. And this kind of helps us move away from the need of fossil fuels for a lot of these products. So I just want to thank you for your attention, and I look forward to seeing you at Northwestern, where you can take weekend bike rides to the Bean and analyze your data with a view of Lake Michigan, as I often do. And if you have any question about my research or life at Northwestern, I have my contact information right down here. Thank you.
Thanks, Natalia. And so now we will welcome Zhajin, who will share some of the highlights of her work. As you can see with those first two presentations, you have people that are working on natural organic materials from very different points of view, but there's also a lot of overlap. Okay, good. There you are. What? Zhajin, you're muted. There you go.
All right, I'll start again. Hello, everyone. My name is Daxian Shen, and I'm from Erica Haman's group. I'm currently a fourth-year PhD student, and I got my bachelor's degree in environmental engineering at Zhejiang University. So the common thing about these two universities is that they are both very beautiful and have great lakes. However, we do get paradox like frozen temperatures here and giant snowmen. I came here majorly because I'm deeply attracted by the beautiful microbial world. This specific paper related to my undergraduate research greatly inspired me by this specific sentence of significance. So it says, our finding opens up the possibility that oxygen was available to microbial metabolism before the evolution of oxygenic photosynthesis. So if their finding proves true, this is going to be a very revolutionary discovery. And I think there are a lot of mystery out there in the microbial world for us to uncover. I want to, besides, I also want to make a tiny contribution to helping the world become more sustainable and help the human being living more healthy. So that's pretty much why I came here. The keywords of my research are molecular methods, pathogen surveillance, and environmental microbiome. The specific environment I focus on for my research project are medical intensive care units. I study the interaction between patient microbiome and environmental microbiome. And I also investigate the effect of chemicals, especially antibiotic chemicals, on the interaction. And in the process, I'm also interested in improving the methods for us to understand this relationship. And our lab studies microbiology in the intersections of public health, built environment, and antibiotic resistance. And here I would like to highlight two projects that are actively running in the lab. The first is CRISPR interference therapeutics, which are led by our new postdoc, Siam. And the second one is the relationship between antimicrobial surfaces and cleaning products. We focus both on more traditional cleaning products like chemical-based products, for example, benzoyl chloride, and newer version of cleaning products like probiotic cleaners. And besides, we also have a couple of projects which are more clinically relevant. For example, Erling Wu, who is currently a first-year fellow in infectious disease, and her research interests include resistant Helicobacter pylori infection, wastewater epidemiology, and the politics of health as they impact access to care. And Shu Chenfeng, who is most interested in how environmental microorganisms, particularly those in wastewater, impact human and public health. And she also launched a statewide wastewater SARS-CoV-2 surveillance project in Wisconsin, and it contributed to the successful real-time surveillance in multiple cities. I think overall, our lab is quite diverse and open in terms of research topics. We mostly focus on bacteria, but we also launched a couple of projects related to phage and other viruses. We use traditional methods, for example, culture. We use a lot of next generation sequencing methods and other molecular-based methods like qPCR. But we also embrace newer technologies like CRISPR. So I think as long as besides microbiology, we also do some work in the intersection of chemistry and microbiology. So I think our lab is quite open and diverse. As long as you have an interested topic, we can always work together to find a topic that suits your interest and the lab's major focus. And with that, I would like to introduce the group, all the postdoc and the graduate members, as well as Dr. Erica Hamman. Thank you.
Thank you very much. All right, and now we will go to our last speaker, Sam Wallace.
All right, let's see. Okay. Does it look good? Do you see a screen here? Excellent. All right. My name is Sam. I am an environmental engineering PhD candidate in my 4th year here at Northwestern. First off, I want to say thank you all for considering Northwestern, first of all, and for your attention here at some point in the future when you're watching this. And I hope to see you all again in a few months when you've all been vaccinated and have decided all across the board that Northwestern is the place you want to continue your graduate studies. So looking forward to that. I want to give a quick background about who I am, just because I was in your position not too long ago. So I got my undergraduate degree at the University of Iowa. I was an environmental sciences major, which is to say I wasn't an environmental engineer at that point in time. But I got a wide range of research experiences in a couple different labs there, including an environmental engineering lab, which hooked me into the environmental engineering research and what led me to pursue environmental engineering here at Northwestern. So I was really interested in these environmental engineering programs, particularly ones that were looking at water quality and water resources, which is what led me to JF's lab, also known as the Laboratory of Metals and Aquatic Systems. I I don't think I really need to make this pitch to you all, but metals, they're critically important from a couple of different perspectives. They're essential for life. They're essential for the technology that brings us together here today. But with these metals, if you get too much of them, if they're in the wrong oxidation state, or if the atoms around these metals are just in the wrong combination, they could be damaging either directly through toxicity, through consumption, or indirectly through environmental damage, which is to say really that speciation matters in this case. So at the center of that for us is a technique known as X-ray absorption spectroscopy, which uses high intensity X-rays to directly probe the coordination environment of elements in a sample. For that, we usually go out to the Advanced Photon Source at Argonne National Lab. That's where we collect most of our data. That really is the centerpiece of our research. And zooming out quickly here, I just want to go over some examples of the research that have been conducted in this lab. So this is an example of using XAS data to inform computational models of the structure of metals in solution. We've also got some work, really fundamental work, looking at the interaction between microbes and mercury, seeing how that affects bioavailability and fate. And more recently, there have been a couple projects with biosensors. This one is a collaboration with people who are just down the hall from us in chemical engineering for a sul-free metal biosensor here. But turning the focus back on myself here, I guess, my focus is more in the broad sense, looking at how metals interact with complicated surfaces. Because in nature, you don't get to work with the super neat, the homogeneous controlled systems that we as engineers really love to create in the lab. When you've got organic matter, you've got minerals, you've got microbes, all mixing together and making up these different surfaces. Oh, my dog's running away here. I'm sorry about that. So I guess when you've got all of these different components coming together, there's a lot of different ways metals can interact with these surfaces. A lot of different places they can stick to these surfaces or sorb to these surfaces. So for this research specifically, the surfaces that I've been looking at are drinking water treatment residuals, which I'll also call DWTRs throughout this presentation. So at drinking water treatment plants, they mix in a pretty wide range of chemicals into the water early on in the drinking water treatment process to remove suspended particles or remove some other contaminants early on in the process, which leaves behind this sludge here. This forms up a pretty high volume. It's a pretty significant nuisance for municipalities to take care of it. Some of them are fortunate in that they can directly discharge it into the environment. They just have a pipe from inside the plant to outside the plant. That's great for them. Other places have to go through a bunch of hoops to apply it to land. But in any of these cases, that's really the end of the useful life cycle for these drinking water treatment residuals. But there's been some really interesting work done that's looking at drinking water treatment residuals as a promising material, as a metal sorbent. So that's sort of the foundation here. In terms of the specific application, we're looking at what if we could dispose these in a way that adds value to them. So we know that there are a wide range of places, sediments that are contaminated with metals like copper and mercury and lead. What if we were to cover those contaminated sediments with a layer of this material, which previous research has indicated is really good at scavenging metals from solution? In this sense, we could use a waste as a resource instead to preserve our environmental health. Now, there's a few questions that you have to answer before you can really dive into, just applying this in the field. First off, is it safe to do this for the environment? Next, is it effective? And finally, as scientists, we want to know, how does this work? So look at this first question, if it's safe. This is a quick look at some bioassays for marine organisms done with collaborators down at the University of Florida to see if organisms can survive and grow when living on these DWTRs instead of sediments. The answer here with these organisms and with these DWTRs is a pretty resounding yes, with no difference between the control and the DWTR samples in either growth or survival here. So this is a really good first step for this sort of application here. But if we want to know more, we should really characterize these samples to find their composition. So I'm going to go into these shared facilities again because they're just so great. We've used a pretty wide range of methods to characterize these DWTR. So I'm just going to show a couple of them here. This one is thermogravimetric analysis paired with mass spectroscopy. It's a good way to get a broad composition of the material. You essentially just heat it up and see what off gases. This instrument is just down the hall from our department at IMCIRC. So what we see here, there's a pretty big difference in between different DWTRs based off of the additives they use. So this is a TGA curve with a lime-based drinking water treatment residual that has calcium oxide added. Most of the mass is wrapped up in carbonate minerals, versus when we have more traditional methods of drinking water treatment, we don't have all that mass wrapped up in carbonates. It's wrapped up in organic matter or water. So that's interesting. That gives us an idea of what might be active in sorption processes with these materials. We can also use X-ray diffraction to get an idea of the crystalline phases that are present in the sample. So these are some examples from the Cohen XRD lab, which is attached to Tech just through a skyway here. But they have some lab source X-ray diffractometers, which works excellently for highly crystalline materials like this here. But if you have some less crystalline materials, it's not quite as great. But we have the option of ramping up to high resolution XRD, which happens at the advanced photon source using synchrotron radiation. From that, we can get a good idea of minerals that are present in crystalline form within all of these drinking water treatment residuals. Finally, just short on time here, but I want to, because talking so much about XAS, I want to give a little bit of data here. So this is principal components analysis of data for copper that is sorbed to drinking water treatment residual samples at a couple of different pHs. So when you just sort of eyeball this with, so these are split up into their principal components here, we can sort of think of this as being in 3 distinct clusters. This can be confirmed with cluster analysis, but I'll just circle them now. So this happens to mesh nicely, these clusters, with 3 different groups of additives that are used at the drinking water treatment plants. So this is lime additives, this has potassium permanganate, and these are the more traditional drinking water treatment residuals. So that's interesting, and I'm hoping that you'll be able to read a lot more about this and a deeper dive into this in the fairly near future here. So just wrapping up, there's some promising results for our ideal application of drinking water treatment residuals here. There's some experiments in the works right now to add some more complexity, looking at the interactions in the lab between actual contaminated sediments and these drinking water treatment residuals. So I'm excited about that. But I really want to emphasize a point that was made earlier, which is these shared facilities here and how great it is to be able to just pop online, reserve time with some world-class instrumentation, and just get the information that you need right away. It's an important piece of my research up to this point. It will continue to be an important piece of my research here at Northwestern, and I'm looking forward to continuing it here. With that, I want to say thank you very much, and I will hand it off yet again here.
Wonderful. Thank you very much, Sam, and thank everyone else for these very illuminating and inspiring presentations. It's remarkable what these students have done in an extremely short time, because I remember all of them on their visit to Northwestern to check out, you know, whether we were a good fit for their graduate study. And I'm so happy that they found out that it was, because they're doing great work. Thank you once again very much for visiting our virtual presentation. Hopefully next year this will all be in person, and we look forward to the presentations that you'll make to us. All right, thanks everyone. Have a great evening, and let us know if you have any questions. Thank you.